know, Adrian is a true pioneer in the field of epigenetics, and he has made many seminal contributions. An early key finding, as you're probably all aware, has been his discovery of CPG islands. These are regions in the genome where the cytosine in the CPG dinucleotide is not methylated, unlike in most of the rest of the genome in mammals. And then this was followed by the quest to identify proteins that bind methylated cytosines. And as you all probably also know, the result of this was the work from his lab, namely the purification of MeCP2 and another number of other methyl cytosine binding proteins. And over the past 2 decades, his work has really been, the efforts in his lab have really been focused and trying to understand the role of methyl cytosine binding proteins and to try and understand how methylated CPGs as well as unmethylated CPGs control the formation of chromatin and regulate gene transcription. And with this, I'd like to just hand over to Adrian and thank you for looking forward to your talk.
Right. Thank you very much. Thanks for the introduction. Thank you for inviting me. It's always a pleasure to come to Cold Spring Harbor. I'm going to talk about CPG as the genomic signaling module, but because in 30 minutes I didn't think I could do a 2-part talk. I realize you guys only have 12, but even in 30 minutes I didn't feel as I could combine 2 topics. So I'm going to focus on one and just give an introduction that's rather broader than that first. And really the star of the show, if you like, is CG, a dinucleotide sequence, which has the properties of a sequencing module. However unlikely that may seem, 2 base pairs seems too short to be biologically relevant. But this one has properties that suggest that it's functioning that way. It's very short, so you can sort of put it anywhere. It's self-complementary. It's one of the 4 possible dinucleotide symmetrical sequences, but it's the most interesting largely because it can exist in 3 distinct chemical forms. Actually, I know there are more than that, but I'm just focusing on 3. One is in the unmethylated state, and the other one is methylated, and the third one is hydroxymethylated. And combinations of those, plus formyl and carboxy, add to this repertoire, so it exists in multiple chemical forms. Specific proteins are attracted or repelled by those different forms, and it varies in frequency very greatly, and this is accentuated by the fact that this form in particular is mutable. And so over millions of years, methylated cytosines, methylated CPGs become depleted and reach a lower level, except in places where there is no methylation for millions of years, such as, for example, CPG islands that are unmethylated in the germline. And there, these lollipops representing CPGs have never been subjected to this mutability, and this dramatically makes dramatically different the density of CPG inside the CPG island and outside. And this gives more information within just the distribution of this CPG sequence itself. So this talk is going to be about proteins that are attracted or repelled by the modified forms, and actually it's going to deal with two really, but predominantly 1. So reading the DNA methylation signal, there are CG binding proteins which can't bind to methylated DNA, and there are methyl CG binding proteins that can't bind to non-methylated DNA, and hydroxymethylation, we're going to hear in the talk afterwards, also plays into this story. So CXXC proteins, such as CFP1, this motif discovered by David Skolnick, binds to non-methylated CG and is repelled by methyl CG. And to sort of summarize in a ludicrously simple cartoon-like form, there is evidence that a sort of sunny promoter-friendly, but also computer-friendly, and abacus-friendly, is environment is created around this CG. And where methyl CG binding proteins bind, they're repelled by non-methylated CG, and when they bind here, one can consider that the data, and some of the data I'm going to show you, fits with the idea that it creates a rather more frosty and hospitable like Edinburgh in June. Or at least this June. So this is obviously a ludicrously simple, and the most interesting parts are nearly always in the detail, but I'd like you to bear these ideas in mind so that you can compare them with what gets said. And I'm not going to say very much about the non-methyl CG binding protein, except that this really is an advertisement for Lizzie Wachter's poster, where she's going to talk about the influence of DNA sequence on CPG islands. She puts in an entirely fictitious, synthetic, CPG-rich chunk of DNA, and she finds, as has been reported before, but in a gene, this is in a gene desert, using a completely artificial sequence. Actually, Brad Bernstein's lab showed it with a piece of E. coli as well, that you get now a new focus of H3K4 trimethylation, about which we've already heard in this meeting. You also get H3K27 trimethylation over a splodge of DNA surrounding this artificial CPG island, and you look at SUSY12, the enzyme that methylates lysine 27, and that's there too. There is no evidence of any RNA polymerase whatsoever. So this is basically what has been called a bivalent CPG island, and I'm not going to go through all the controls, which is not really poised to do anything. It's just, if you like, the default state of a piece of CPG-rich DNA. It's what the DNA does to the chromatin. And I think that's an interesting view, and maybe you will visit Lizzie's poster, and that's just a control showing histone H3. So I'm not going to say any more about that, but there is obviously a whole story about the influence of CPG density on CPG island function that I'm not alluding to here. Instead, I'm going to talk about a protein that binds to methylated DNA, namely MeCP2. And I'm always suspicious of people who show front pages of their papers. It sort of implies they feel they've been wronged in some way, and they want to rectify it by showing all that time ago that they published something that was very prescient. But it's really up there because we found MeCP2 in 1992, and far from illustrating my prescience, I want to make it clear that you may well consider that 20 years is a long time to study one protein and still not be entirely sure what it does. But fortunately, this is a chromatin meeting, so everybody's more or less in the same position. So this is how we found this protein, by running all the nuclear proteins on a gel and then probing with methylated or unmethylated DNA. Found a protein that bound to methylated DNA. This is an X-ray structure showing the DNA binding domain associated with the methyl groups. We know how it does it. But I'm going to talk about the way in which our ideas about this protein have fluctuated and changed with the arrival of, and become more sophisticated, I would like to think, with the arrival of new data. And a major motivation for this study is the finding by Huda Zogby's lab that this syndrome called Rett syndrome is caused by mutations almost exclusively in the MeCP2 gene. This is taken from YouTube, and it's a girl with Rett syndrome. It's a profound disorder, affects 1 in 15,000 girls, not that common, not that rare either. And it has these various features, which you can see illustrated there. Breathing abnormalities are fundamental. There are others, digestive abnormalities. Lifespan approximately 40 years on average, during which more or less 24-hour attention is required. So there's this sort of, I think originally people were disappointed that here was this gene, it's expressed everywhere, MeCP2, and MeCP2 is a protein that is binding to methylated DNA. And it's expressed everywhere, and it wasn't really a nice brain gene. It was a housekeeping gene, if you like. We now know that neither males nor females, or we've known always that neither males nor females with MeCP2 mutations survive to reproduce. They survive, but they don't survive to reproduce. And so all these are new mutations. And so you get a huge spectrum. And really the informative part of the talk is that the mutations are telling us an awful lot that we hadn't previously exploited as well as we should. You can see there throughout, and you can find papers which say that MeCP2 is mutated throughout its length. And indeed, that's what it looks like. So Rett syndrome patients are mosaic for MeCP2 expression because the MeCP2 gene's on the X chromosome. So due to a mosaic X chromosome inactivation, either the MeCP2 plus gene, the wild-type gene, is inactivated, and this is a MeCP2 negative cell, or the other way around, in which case it's essentially functionally a wild-type cell. And it's the mosaicism of these two cell types in the single brain that cause the disorder. And this is a piece of a mouse brain, a dentate gyrus, just showing holes in the MeCP2 pattern that show up as red. This is DAPI here. And this is a clone of cells that's derived from MeCP2 negative progenitor, and this is a clone derived from an MeCP2 positive progenitor. So although one thinks of X inactivation as generating gigantic patches on tortoiseshell cats on the skin, in the brain the patches are really tiny and interspersed with each other. So you can make a model. We made a model a long time ago. Rudolf Jenisch made a model at the same time, and it produces a phenotype that in males is severe and leads to death, just as in humans, whereas in females, it leads to a late onset chronic phenotype that has a lot of features of Rett syndrome. So the animal stops moving, it's not breathing rhythmically, it's breathing then stopping and breathing and stopping, one of the features of Rett syndrome. It's very low to the ground, and it has these tremors, and when you pick it up rather unceremoniously, instead of putting its hind limbs apart, as a wild-type mouse would do, it clutches them together. And then another feature that arose in this aspect of our work was that if you put MeCP2 back, and that actually it's in the same mouse that's shown on the left, then MeCP2, then the mouse which would have had to be killed within a week or so, at this stage, this mouse becomes this mouse and is remarkably restored. And so that surprisingly, you put back the missing piece of the machine and the machine works, which doesn't sound all that surprising at all unless you are a neuroscientist, in which case it's a surprise. In fact, we all think, I think, of neurological diseases as permanent. So Rett syndrome in mice is a reversible condition, and reversible, this obviously has therapeutic aspects, but I want to think about it in terms of the function of you to think about it in terms of what MeCP2 might be doing. So these are some of the early conclusions about what MeCP2 was doing. It binds methylated DNA. It associates with a histone deacetylase complex, and the deacetylase complex we associated with was a SYN3A complex. It mediates transcriptional repression in artificial systems by transfection, and our hypothesis, therefore, was that the absence of MeCP2 causes derepression of methylated genes. Fine. Then you look in the mouse brain that doesn't have MeCP2, and you find modest, a whole host of changes, but very modest, and genes go up and down. So this didn't really fit with the simple repression model, which is shown here as a recruiter of HDACs. And within the sort of confusion that followed that about the paradigm, we have the model that it is involved in chromatin compaction, irrespective of DNA methylation, that it's a transcriptional activator, recruiting CREB, that it's involved in modulating alternative splicing. This one is not heard about so much now. And chromosome looping. There's one figure in one paper that suggests that maybe it can do that. So there are lots of hypotheses out there, and so it's necessary to go back to the drawing board and look more closely at MeCP2. Does it target specific genes? Is chromosome binding really the important thing, or are we misleading ourselves? Because of the way we found it, we're looking at certain types of routes. And does it cause transcriptional repression? Well, back to the mutation picture, what you notice is that there are missense, nonsense, frame shifts, and deletions, And the blue ones are missense. And really, they're the only ones where the mutation is at the place where the functional problem arises. In the other cases, everything downstream is destroyed. So what Matt List did is, who is here, and has a poster, is look at our Rett, I think he has a poster, Rett syndrome missense mutations that are not found in the parents. If you go to the database for MeCP2, you find polymorphisms, you find all sorts of stuff. To single out the ones that were causal, he looked at ones where the parents had been tested and they didn't have the mutation, so the mutation arose anew in the patient. And what you now see is two clusters, really. A cluster over the DNA binding domain and a cluster up here. And interestingly, I found this database where you can find polymorphisms, which these are people who don't have Rett syndrome, 6,000 exome sequences. And what you see is that there are mutations spread throughout, but they jump over the two regions that are involved in causing Rett syndrome, the DNA binding domain and this mysterious cluster here. So this is the reciprocal of the mutations that cause the disorder. So I'm going to first of all look at the DNA binding domain, because there's quite a lot of evidence that this DNA binding domain binds methylated DNA. There's a crystal structure. You can use it as a reagent to purify methylated DNA from non-methylated DNA. Tony Kouzarides showed methylated chromatin pulls out MeCP2, whereas a non-methylated doesn't, binds preferentially to methylated alleles in vivo. If you do CHIP, it pulls down the methylated allele when both are present in the same cell. It tracks DNA methylation by CHIP, and many MBD mutations that cause Rett syndrome abolish binding. But nevertheless, there is still a sort of suspicion, I think, that maybe it isn't really a DNA binding protein, even after all that evidence, and certainly no sorts of evidence that one can produce are ever totally watertight. So the best thing you can do is stack all sorts of different evidence, one on top of the other, to improve the probability that you're right. And so this just shows, first of all, that MeCP2 is extremely abundant in neurons. The number of molecules, nearly 20 million per nucleus, in neurons is really astronomically high. It's nearly as high as the number of nucleosomes, as the number of the methyl CPGs in the genome. And so there's enough to coat the genome. So this is an extraordinary finding, really, that just by measuring the abundance, you learn something about MeCP2. There's enough to coat the genome. And indeed, this is a whole chromosome, so it's ludicrously low resolution. But this is CPG along here. This is in glia. And you see CPG goes up and down along the chromosome, and MeCP2 is tracking it. And remember, the vast majority of, well, the majority of CPGs are methylated. So this is basically tracking DNA methylation, and it looks that way in detail as well. There was a poster from Dirk Schubler's lab. I'm sorry, I've forgotten the name of the person I spoke to for a long time about it, which supports the fact that it's binding methylated DNA. So MeCP2 does not behave as a transcription factor with specific target genes. It's all over the genome. So what happens to MeCP2 binding in a DNA methylation deficient brain? Actually, Rudolf Janisch made a conditional knockout of DNMT1 and knocked it out in neurons and glia using nestin-Cre. And the animals were born, and then they died instantly because they couldn't take a breath. But actually, what Sabine Lager and Christian Seiser and terrible memory for names, but she's here and she has a poster, and I hope you went to it today. And I apologize to her in person afterwards, have found is that if you cross this DNMT1 mutation onto an outbred background instead of an inbred mouse, which is rather more vulnerable, you can get survival. These animals will survive for a reasonable length of time, and now you can look at MeCP2 binding in the brain that doesn't have DNA methylation. So here's the red stain shows that DNMT1 is missing in these brains, even though it's present everywhere else, and DNA methylation by a simple restriction assay is reduced. And, but MeCP2 is there. This is a postnatal day 5. in the wild type and the knockout, and these are littermate pairs, and this is in postnatal day 12. There is MeCP2 in comparable amounts in these two sorts of animals, but if you do CHIP on intracisternal A particles, again in littermate pairs, wild type, knockout, wild type, knockout, wild type, knockout at postnatal day 12, there's very little binding. And the same at a single copy gene, brain-derived neurotrophic factor, wild type, knockout, wild type. These are individual mice. So basically, whatever it is that MeCP2 binds to depends upon the initial event of DNA methylation. So does MeCP2 target specific genes? No, it really doesn't make that much sense to talk about target genes necessarily because it's all over the genome. In fact, there's almost enough for every methyl group, particularly if you consider half of them are facing the wrong way. So is chromatin binding dependent on DNA methylation? Yes, it binds a 2 base pair sequence that occurs once every 150 base pairs in the genome. So does MeCP2 inhibit transcription? Well, the data on this is, as I've said, is rather unclear simply because of what happens when you take it away. Some genes go up, some genes go down, bit of a mess. Does MeCP2, so what is shown by 3 different labs is that a histone acetylation is elevated in the MeCP2 null brain. So even in the whole organ, you can detect changes in histone acetylation. So now that brings us to this region here. What could this be doing? This is a hotspot for mutations in Rett syndrome. And if you look at the so-called, I've got the classical view here, but it's just the old view of MBD transcription, which is the methylated DNA binding domain, the transcriptional repression domain defined by model experiments. Now, if you look where the Rett mutations are, there's mutations there, there's one there, about which I'm going to say nothing unless anybody asks me, and this is the methyl binding domain. If you look at evolutionary conservation, and now you have vertebrates going right the way back to lamprey, I will mention in passing, nothing like MeCP2 is found in any invertebrate, even though they have what pass for brains within them. So this is not a ground floor of the brain evolution protein. It's more or less a vertebrate add-on. And then we can ask now, what do we know about these domains? This we know, as I've said, is DNA binding domain. There's an AT hook here, but this isn't mutated in Rett syndrome. We see a nuclear localization signal. That's not mutated in Rett syndrome either, interestingly. I think missense mutations aren't enough to destroy a nuclear localization signal, and there may be secondary ones anyway. But then that leaves the question of what's this? This is the hotspot we're interested in. One possibility is it's a protein interaction sequence, and like most proteins, the number of candidate interactors is proportional to the number of labs that work on it. And this is MeCP2 interacting proteins. Some of these are probably real, others less so. And none of them has been shown at this point to be affected by mutations that cause Rett syndrome, which is the critical thing. So Matt decided to use techniques that are not exactly earth-shattering, but had not been used before, and he put it together in a logical way. So to use nuclease instead of high salt to make the extracts, to use, because we've been making standard dignum extracts, mouse brain with a knock-in of EGFP fused to MeCP2 made by Jim and Jackie in the lab, and this meant that he could use this single-chain camelid antibody, which is very clean, small, discrete, high affinity, and do the experiment. And what comes down is MeCP2, thank goodness, and then 2 important subunits, which we know from Rob Eckert's work now bind to the nuclear localization signal, and 5 subunits of NCOR. NCOR was on the list that I showed you before, but we had previously thought that SYN3 was the complex that was associated with MeCP2, but no mutations seemed to destroy that interaction, and that's still the case. No Rett mutations destroyed that interaction. But now we're looking at this complex here, which is traditionally associated with recruitment by unligaded nuclear receptors. Again, a histone deacetylase complex has HDAC3, which is only active when it's in the complex. And this is simply a set of experiments to confirm that interaction by CHIP. The native protein without GFP also brings it down. The region that is responsible for the binding is very discrete, and it coincides with that cluster of mutations that I introduced earlier. And indeed, if you, these are co-transfection experiments. If you co-transfect with these Rett mutations, 306C, 305R, 304E, 302R, then you abolish the interaction with these two components of NCOR, but not with A2979V. So this, the map, we think we have the interactor for the upstream region. So we made some knock-in cells with the two most common Rett mutations, T158M and R306C, together account for about 15% of all the mutations. That's partly because they're at CPGs, which are mutable, but those are the blue ones there. We made a knock-in for each of those. ES cells, converted them into embryoid bodies, turned those into neurons, and what you find is that for T158M, localization to the heterochromatic blobs, where most of the methyl C is fine in the wild type. It's fine in the, so these dots are fine in the 306C mutation, which is here, and it's not fine in the T158M because this is a mutation in the DNA binding domain. But conversely, if you look at interaction with NCOR SMART, you find that it's abolished in the 306C, but DNA binding, as I've shown here, is fine, and it's present in T158M. So basically, we have two regions of, and this just shows that SYN3A is not affected by these mutations. And if we do a transcriptional repression assay, we find that the region, the N-terminus doesn't repress at all, but the C-terminus represses. This is fold repression, a slightly backwards way of showing it. And if you put in TSA, inhibitor of histone deacetylases, all the repression goes away. The same with this other mutation outside that region, but all the mutations in the region more or less abolish the repression. So these mutations that destroy the interaction with NCOR also destroy the ability of this protein to do transcriptional repression. So in other words, we have a model like this, and this is a model, it's a cartoon even. This is MeCP2. I'm showing it in its, as an abundant protein that's all over the place, binding in a DNA methylation-dependent manner. So this is in neurons, for example. And then we know that NCOR is much less abundant. So it's not a one-for-one thing. This is not a stable complex, but it's a good interaction. It's a strong interaction that's easy to detect. But then we sort of imagine that NCOR SMART comes in and maybe removes an acetyl group and then disappears, and then another one comes in. So it's sort of a platform for it to appear. This is now exhausted all my PowerPoint skills. There it goes, I bounced. I was going to have histone H1, which I haven't mentioned, being displaced. MeCP2 displaces histone H1. When it's missing, you get twice as much histone H1 in neurons. But unfortunately, I couldn't. If you can just close your eyes and imagine histone H1 disappearing when MeCP2 appears, I'd be grateful. So, this is all fine. This is a recruiter of a histone deacetylase complex. What transcription is modulated by MeCP2? Is it specific target genes? There's no evidence for that so far, although people have published papers about target genes, including us early on. Transcription in response to neuronal activity. Mike Greenberg's lab has shown that MeCP2 is a phosphoprotein. It is, changes its phosphorylation status at specific sites when neurons are stimulated. Maybe this is something to do with it. And this hasn't been assayed properly in terms of what happens when MeCP2 is missing or compromised. And a third possibility is rather boring and transcriptional noise, but there are several senses in which one can use noise. The sense I mean here is that it's inappropriate transcription. So, for example, Pete Skeen a few years ago showed that there was a reproducible increase in the expression of L1 repeats, major satellites, and IAP elements in MeCP2 deficient brain. You only saw it in the nuclei. You couldn't see it if you took the whole cells. And there's another experiment here, which is a model from Fred Gage's lab where transposition of a retrotransposon leads to its fluorescence with GFP. And so in a wild-type animal, you consistently see much less transposition than you see in MECP2 knockouts. So consistent with the idea that MECP2 is acting as a repressor of some kind. But it still is not satisfying. We have no satisfactory explanation at this point as to why this global moderate dampening effect on transcription is required, or under what circumstances it is required by neurons. And that's extremely irritating, and we're obviously working very hard to address it. So what we know about MECP2 is it's a vertebrate invention. There's nothing like it in invertebrates, and I've spoken to evolutionary biologists who look for orthologs of this kind. It's expressed in most tissues, but it's extremely abundant in the brain, particularly in neurons, but also pretty abundant in glia. It binds chromatin in a DNA methylation-dependent manner, and it fights with histone H1 and displaces it. In fact, the N-terminus of MECP2 rather resembles histone H1 in some respects. So, of course, saying something that something does the same as histone H1 doesn't get you all that far in understanding what it does, but it's nevertheless an interesting parallel. It bridges between methylated DNA and the NCOR SMART co-repressor complex and missense mutations that break one or other of these contacts, either end of the bridge. That's what causes Rett syndrome. The whole protein is actually very highly conserved, particularly among mammals. But that doesn't matter. That means it is doing something. But in terms of Rett syndrome, only those two bits are important. So is disruption then the exclusive cause of Rett syndrome? Is there anything else? And this is the last slide. It fits. So in our argument for this simple idea that it's just a bridge and either end of the bridge is what, loss of either end of the bridge is what causes Rett syndrome, it fits with where the missense mutations are, most of them. It's simple, so it's attractive for that reason, although not everybody goes for simplicity, but it beats complexity in my opinion. But, and against P225L and P322L, two mutations that they're not in the right place. They are both prolines, so they may well change the structure in ways we don't understand. And the C-terminal deletion, this is the biggest problem. Those red things there, that's the chopping off of the C-terminus. And this would leave, if this protein is stable, both of those domains intact. So predictions of the simple model are that these latter mutations, that's these two and all these, either destabilize the protein or they cause steric interference with one or both of these critical domains. So these are testable hypotheses. If they're wrong, then it means there's something we're missing. But if by any, so we're obviously interested in testing that. So I would like to finish by acknowledging the people who've been involved in this. Matt List did the NCOR discovery, Jackie Guy, Jim Selfridge made the mice that were crucial for this. Rob Eckert did a lot of the transcriptional analysis. Sabina and Astrid, Hagel Kreuz and Christian Seiser were involved in looking at the DNMT1 mutations. That's the person whose name I forgot earlier. And Elizabeth Wachter has a poster here, and Bernie Ramsahoy allowed us to analyze the DNA methylation levels in those mutants. And these people funded the research. Thank you very much.
Here's one. You showed that two lysins were mutated in the Rett syndrome, the 304 and 5. So did you ever test if there are some post-translational modification that may well be responsible of recruiting the N-Nod complex? Like
you mean, could the mutations in the lysines in that region be because of lack of modification?
Yes. Yes.
Well, I mean, it could, but they're in this domain. The most parsimonious assumption is since they're in a domain, several other amino acids within which several other amino acids destroy the interaction. I mean, I can't argue against what you say, but you know, we're trying to determine the structure of that interaction, and that'll tell us what every amino acid does. There is no evidence at the moment that those lysines are modified.
Adrian, I wondered if the location of the corepressor complex co-locating with MeCP2 will help you computationally to find other DNA binding proteins, et cetera, that might direct you to a specific function at the, you know, at those locations, transcription of those.
Yeah. I mean, if you do chip with MeCP2, it's all over the place. And
The location of the corepressor.
the NCOR, where the NCOR is, you mean you think it might select certain regions to visit and discriminate against others? Yeah. There is a problem we're having with that, in namely that there are two lysines very close together. I think they're the ones you're talking about. These seem to cross-link almost immediately and prevent binding. So we're actually having trouble doing the chip because the cross-link process is faster than the intramolecular cross-link is faster than the intermolecular. But that's our interpretation of what we're finding. But yeah, you might well be right. We're worth doing.
Adrian, if MeCP2 is a bridge, would you expect to find mutations in Rett in NCOR?
Well, the only reason that would be rare is that MeCP2 is on the X chromosome, and that's why it's so abundant. For the NCOR, because it's not on the X chromosome, its components are not on the X chromosome, you'd need both alleles to be mutated. And that would be very rare, but the answer is yes, you would, provided there is only one contact point on NCOR for MeCP2 in one of the subunits, and that may not be the case.
Are the phenotypes absolutely identical in the two portions of the protein, TDR and in the middle binding domain?
Yeah.
Or are there
so
differences
they're
between?
all, people have, obviously when the gene became known, when Huda Zoghbi showed what the gene was, people tried to do genotype-phenotype correlations. It's very, very difficult because the phenotype it's always severe, but it's quite variable. And it's been very, very difficult to do genotype-phenotype correlations. One of the slightly milder mutations is one you'll hear about in the next talk. The C-terminal ones tend to be milder as well. They're probably the mildest, the ones, the red ones near the end.
No, I'm talking about the two bridge points essentially.
The two bridge points, they're not really
Indistinguishable.
distinguishable. Yeah, they both cause exactly the same phenotype. So I think that the mutations are telling us the important bits of the protein, and now we know what interactions of both those. That simple model is that's all there is to it, but simple models aren't always right. But I think this one stands a good chance.
How good do you think the mouse model is? You can cure
well, that's always the problem. I mean, the mouse model is, you know, it's late onset in the females. It's chronic. It's breathing problems. It's tremor. It's limb clasping. It's, you know, obviously failure to speak and walk is not something one can necessarily assay. But, you know, as these models go, it's pretty damn good. If you compare it, say, with Fragile X syndrome, Fragile X mouse, the FMR1 knockout mouse, is viable, fertile, breeds fine. You do behavioral tests and you can find things. Rett syndrome is much more overtly like the syndrome, according to most people. But of course, that doesn't mean that a therapy in the mouse would work in the human, because there's lots of scope for differences to everything. Therapy. Well, people are trying all sorts of things, everything from gene therapy to reactivating the silent X chromosome. I mean, that's what, if you guys want to do something, that would be something. Digging out the gene on the silent X, what's the point of doing gene therapy when there's already a good gene in there? They're heterozygous. Switch it on. But we have no idea how to switch on a silent gene on the inactive X. So that would be a chromatin option. And other people, there are other things. There's an IGF-1 trial. There's all sorts of stuff going on. So in a sense, what the reversibility did was catalyze the idea that you don't have to diagnose this before there are any symptoms. It's potentially reversible after that. And so there's a lot of activity in that area.
Can you further comment on why you think this is reversible when it's affecting the genome in a global sense?
Well, I know epigenetic things, I mean, epigenetic things are supposed to be forever because we're led to believe that epigenetics is forever. Even though it's not genetic, it's nevertheless long-term. Actually, we're seeing a disturbance in the epigenome here. You put back the protein that's missing, and the epigenome does as it's jolly well told. It goes back to where it was before. And I think there is, in general, to answer at perhaps higher philosophical level, if I can call it that, than you're asking, I think that the epigenetics more or less does what it's told, in particular by transcription factors. And you know, the idea that the epigenome gets changed and then it's the devil's own job to make it go back again is not necessarily right. It's really rather plastic.
Hi. Have you tried to knock in the Rett syndrome-derived mutants into the mouse model rather than just the knockout and see if it has any different phenotype?
We have T158M mice, indistinguishable from the knockout. We have 306C ES cells, which I showed up there, and they're in chimeras in our lab, and we have a collaborator who also has them in chimeras. So this is on the way. I think you're right. This is the way one wants to go, to actually use the mutations, reconstruct the mutations, rather than just say the knockout is the model.
So I think we better probably move on to the next speaker. Thank you, Adrian. So